Hi and welcome to my OCR A-Level Biology revision with me, Christine. So today's lesson we're going to look at genetic code and transcription. So the genetic code then is when we talk about our DNA sequence. So our DNA-based sequence codes for the sequence of amino acids, and therefore the genetic code is that DNA sequence of those bases. So it's important that we note these key definitions that you will have to refer back to. So your DNA codes for the sequence of amino acids, that primary structure of your polypeptide. Now, we know that our genetic code comes in what's known as a triplet code. So there are 3 DNA bases that code for one amino acid. So as you can see here, my thymine, adenine, cytosine, if that is the sequence, that will code for the methionine start codon. So the next thing we need to understand is that these triplet codes are non-overlapping. Not only are they non-overlapping, but they're also universal. And that means that every single organism uses the same code. So whether we're talking about a prokaryote, whether we're talking about an animal, a plant, a fungi, or a protoctista, the genetic code is universal and therefore it can be transferred into other organisms. So do check out my video on Module 6, manipulating genomes, to find out more on that. Now, the last word key definition you have to know is that the genetic code is what's known as degenerate. And what that means is more than one triplet can code for a specific amino acid. So in this example, guanine, guanine, adenine codes for the amino acid proline, well, so does guanine, guanine, thymine. So it's important to note that the degenerate nature of the genetic code is that more than one triplet will code for a specific amino acid. Now, there are 20 different amino acids, and yet there are 64 possible combinations of our triplet code. And that's why we know that it is degenerate and more than one triplet code will code for a specific amino acid. Okay, so let's look at transcription then and the process of transcription. So we know that our DNA molecule is a double helix. We know that it is enclosed within our nucleus. So transcription is basically the way in which we can take the genetic code, for example, a gene, and we can turn that into a smaller molecule which is able to leave the nucleus. So to be able to do that, we need to take a step back and look at DNA helices. We know that our DNA bases are forming hydrogen bonds between the nitrogenous bases. So we need to unzip and unwind that. And that happens with DNA helices. So the DNA helicase will unzip the strand at what's known as the start codon. So it's important that we remember that the genetic code, the triplet code, can actually be referred to as a codon. So once helicase has unzipped the DNA double helix strand, what that then means is free nucleotides. Now this time they're not DNA nucleotides, they are free RNA nucleotides. So the first thing we need to know is they are RNA nucleotides, therefore they have the ribose sugar. They're going to form complementary base pairs. So cytosine is going to pair with guanine, and adenine this time will pair with uracil because RNA molecules do not have thymine. Instead, they have uracil. So any place where there is an adenine with that complementary base pairing, uracil will pair with that and form the 2 hydrogen bonds. Now, it's important to know of the double helix strand, there's only one of the strands which acts as a template. So that's known as the antisense strand. So that is the antisense strand because it is going to complement the code. The sense strand, which is the other strand, that is going to be exactly the same as my mRNA strand, except wherever there's a thymine in the DNA strand, there will be a uracil in the mRNA strand. So it's important you note, are they giving you the example of the template strand? The template would be anti, so the opposite of what the mRNA strand is, or are they giving you the sense strand, which is exactly the same, except thymine is converted into uracil when it's the mRNA strand. Okay, so we have had our free nucleotides form hydrogen bonds with their complementary base pairs. And then what happens is our RNA polymerase, be careful because we're doing transcription and because we're looking at RNA molecules, this is called RNA polymerase. But it does exactly the same thing. RNA polymerase is going to bind at the promoter region. It's going to move down that gene and it's going to build the phosphodiester bonds on what's known as a single strand of pre-mRNA, messenger RNA. So what we've done is we've taken that genetic code, that sequence of those DNA bases, we have copied them onto this pre-mRNA strand. Now, before we go anywhere, we need to actually modify this slightly. So our RNA polymerase is going to form those phosphodiester bonds and we're going to transcribe and end up with our pre-mRNA. Well, now what we need to do is we need to actually understand that in that pre-mRNA, in that section of a gene, there are areas which are called exons. Now, those exons are known as coding regions. Those coding regions will result in the translation of a polypeptide. The other sections, these lighter pink sections, are known as introns. Now, they are non-coding regions. So introns do not get translated. They do, however, get transcribed. So when we are looking at a gene is made up of both these coding and non-coding regions. So the coding is known as exons, the non-coding is known as introns. So this is what's known as pre-mRNA, and you can see here that I have got 5 exons. Well, what we would then have happen inside the nucleus, we're still inside the nucleus, we haven't left yet. Inside the nucleus, the introns, these non-coding sections of DNA, are going to be spliced out. Now, they're spliced out of the pre-mRNA to leave what's known as mature mRNA. Now, that mature mRNA is small enough now to leave through the nuclear pores. But it's important that we note that not only can we splice together the exons in the way of 1, 2, 3, 4, 5, just cutting those introns out, but what we can also do is we can splice together different combinations of these exons. So it could result in a 1, 2, 4, 5, it could result in a 1, 3, 4, 5, a 1, 3, 5. The way in which they are spliced together, you don't need to know how it happens, you need to know the name splicing. But the key thing is that it can result in different polypeptides being produced. Depending on how it is spliced together to make that mature mRNA will result in a different polypeptide being translated at the end. So the other thing to note is we also remember this mature mRNA is going to leave the nucleus and is going to move into the cytoplasm. Well, therefore, that could result in degradation of the mRNA. So to prevent that degradation and to aid it with its binding onto the ribosome, what happens is before it leaves, it has a cap and a tail added to it, and that helps it to be stabilized so it can make its way to the ribosome for the next stage, which is translation. So I hope you've liked this video, and if you have, please do click the like button below and subscribe to my channel. Also, do check out my revision platform, www.aiqchat.com, to help you with your revision.